Biologist John Craig Venter, better known as Craig Venter, might be one of the coolest and most revolutionary scientists you've never heard of. A pioneer in genetics and synthetic biology, he's constantly striving to make more advanced synthetic organisms and find ways to synthetically support life. He's best known for his first draft in sequencing the human genome and transfecting a cell with a synthetic chromosome. Venter was born in Salt Lake City, Utah. In his youth, he didn't take his education seriously, preferring to spend his time on the water in boats or surfing. According to his biography, A Life Decoded, he was never said to be a terribly engaged student, having C's and D's on his 8th grade report cards. Venter considered that his behavior in his adolescence was indicative of attention deficit hyperactivity disorder, or ADHD, and later found ADHD-linked genetic variants in his own DNA. After graduating from Mills High School in Millbrae, California, his father, John Venter, passed away at age 59 of cardiac arrest. This gave Venter the awareness of his own mortality. He's quoted saying, "If you want immortality, do something meaningful with your life." Though he opposed the war in Vietnam, he was drafted and enlisted in the U.S. Navy, where he worked as a hospital corpsman in the intensive care ward of a field hospital. During his time in Vietnam, he actually attempted suicide by swimming out into the ocean, but changed his mind more than a mile out. Being confronted with severely injured and dying Marines on a daily basis instilled in him a desire to study medicine, although he later switched to biomedical research. After touring in Vietnam as a Navy corpsman from 1967 to 1968, Dr. Venter began his formal education. He first earned a bachelor's degree in biochemistry and a PhD in physiology and pharmacology from the University of California at San Diego. He was then appointed professor at the State University of New York at Buffalo and the Roswell Park Cancer Institute. In 1984, he moved to the National Institute of Health campus, where he developed the Express Sequence Tags, or ESTs, a revolutionary new strategy for rapid gene discovery. While an employee of the NIH, Venter learned how to identify mRNA and began to learn more about those expressed in the human brain. The short cDNA sequence fragments Venter discovered by automated DNA sequencing, he named Express Sequence Tags, or, as previously mentioned, ESTs. Wanting to advance healthcare, Venter became ferocious in his passion about the power of genomics. Venter believed that the shotgun sequencing was the fastest and most effective way to get useful human genome data. This technique determines the DNA sequence of an organism's genome. The method involves randomly breaking up the genome into small DNA fragments that are sequenced individually. The method was rejected by the Human Genome Project, however, since some geneticists felt that it would not be accurate enough for a genome as complicated as that of humans, and that it would be logistically more difficult, and that it would cost significantly more. In 1992, Dr. Venter founded the Institute for Genomic Research, a not-for-profit research institute where, in 1995, he and his team decoded the genome of the first free-living organism, the bacterium Haemophilus influenzae, using the whole new genome shotgun technique he had invented. Venter viewed the slow pace of progress in the Human Genome Project as an opportunity to continue his interest in trying his shotgun sequencing method to speed up the human genome sequencing. So, when he was offered funding from a DNA sequencing company to start Celera Genomics in 1998, he seized it. The company planned to profit from their work by creating genomic data to which users could subscribe to for a fee. This research culminated with the February 2001 publication of the Human Genome in the journal Science. Cool side note I'd like to mention, he and his team at Celera also sequenced the fruit fly, mouse, and rat genomes. Unfortunately, Venter was fired by Celera in early 2002. According to his biography, Venter was fired because of a conflict with the main investor, Tony White, specifically barring him from attending the White House ceremony celebrating the achievement of sequencing the human genome. In 2005, Venter co-founded Synthetic Genomics, a firm dedicated to using modified microorganisms to produce clean fuels and biochemicals. In May 2010, a team of scientists led by Venter became the first to create what was described as synthetic life. This was done by synthesizing a very long DNA molecule containing an entire bacterium genome and introducing this into another cell. The single-celled organism contains 4 watermarks written into its DNA to identify it as synthetic and to help trace its descendants. The watermarks include 1. A code table for the entire alphabet with punctuations, 2. Names of 46 contributing scientists, 3 quotations, 4. The secret email address for the cell. On March 25, 2016, Venter reported the creation of SYN 3.0, a synthetic genome having the fewest genes of any free-living organism, which in this case was 473 genes. Their aim was to strip away all non-essential genes, leaving only the minimal set necessary to support life. This stripped-down, fast-reproducing cell is expected to be a valuable tool for researchers in the field. In August of 2018, Venter retired as chairman of the board, saying he wanted to focus on his work at the J. Craig Venter Institute, which he founded in 2006 as a non-profit which conducts research into synthetic biology. He said that he will remain as a scientific advisor to the board. Today, Dr. Venter and his team continue to blaze new trails in genomics, with him saying he has no plans on retiring. They have sequenced and analyzed hundreds of genomes and have published numerous important papers covering such areas as environmental genomics, the first complete diploid human genome, and of course, the groundbreaking advance in constructing the first self-replicating bacterial cell using synthetic DNA. As for his life outside the lab, he has a home in La Jolla and a ranch in Borrego Springs, California, as well as homes in 2 small towns in Maine with his third wife, Heather Kowalski. He indulges in 2 passions, sailing and flying a Cirrus 22 plane, which he calls the ultimate freedom. And I have to say, that is the coolest thing that I have ever heard of a scientist doing outside of his lab. Dr. Venter is one of the most cited scientists and the author of more than 280 research articles. His most recent paper was actually a research review made in June of 2024 called Physiological Basis for Xenotransplantation from Genetically Modified Pigs to Humans. Quite a mouthful. In which he and the other authors of the paper summarized and analyzed the methods currently being used to transplant pig organs into humans as a way to expedite the organ transplant waitlist. The last research paper he wrote in which he actually took part in experimentation, was made in April of 2019. This paper is called Functional Characterization of 3D Protein Structures Informed by Human Genetic Diversity, and explores how variation in humans' genetics influences protein structure and function. This is important because it highlights how different individuals may respond to diseases and treatment. The authors of this paper, Dr. Venter among them, use various techniques to analyze 3D protein structures. They utilize genomic analysis using techniques like whole genome sequencing or genotyping to link specific mutations with protein structures. They also utilize a lot of computer modeling software in order to 3D model certain proteins. This approach to protein analysis and linking to mutations can help identify potential indicators for disease, allowing medical professionals to tailor personalized treatment plans to patients. As for his awards and accolades, he's also the recipient of numerous honorary degrees, public honors, and scientific awards, including the 2008 United States National Medal of Science, the 2002 Gairdner Foundation International Award, the 2001 Paul Ehrlich and Ludwig Darmstädter Prize, and the King Faisal International Award for Science. Dr. Venter is a member of numerous prestigious scientific organizations, including the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Science, and the American Society for Microbiology. So I'm coming to you live from my dorm room here. This is just how I wanted to wrap up the video. I just kind of wanted to share some personal thoughts. Honestly, I wish that Dr. Venter was talked about more. You know, going into research for this video, I didn't know much about him and thought his accomplishments were sort of cool. But as I researched more and more, I found out about his life and it began to inspire me. He didn't grow up particularly academic and he wasn't gifted in school like many people think that famous scientists have to be. I mean, he was a below average student. He preferred to do other things besides school. I can relate to that. It wasn't until he was morbidly inspired by the Vietnam War that he became involved in academics. And, you know, I think the main reason that this inspires me is because it makes me feel better knowing that it's okay to come from humble and even lackluster beginnings and still be a good scientist or doctor. I feel like this alone makes me identify with him. And on top of that, his research has benefited medicine in such a massive way through his sequencing of the human genome. Plus, his research into synthetic life may hold the answers for a large advancement in modern medicine. Craig Venter is such a monumental figure in modern science, and in my opinion, I think he needs to be discussed more in school settings. His accomplishments, they can't be ignored, and his journey to get to where he is now is truly incredible and inspiring to someone like me. To me, John Craig Venter is my biggest inspiration in the field of biology and, one that I think I'll continue to hold in high regard throughout the rest of my academic career.